products, and that's why even one gene can give rise to multiple different proteins. Okay, and then just again in the form of a review, we did also discuss briefly the process of reverse transcription and RNA, templated RNA synthesis, these two oddballs that embellish the central dogma. But now we're just going to focus on this last step right here, how to take an RNA and turn it into a protein. So how to do that? Well, we talked last time, we have 20 amino acids. We're going to need to use at least 3 letters of our nucleotide alphabet to encode those amino acids. And what are the implications about the fact that there is degeneracy in the genetic code? So that phrase should mean something to you already, degeneracy in the genetic code. And we'll take a look at exactly where that degeneracy lies. So we do need to find a way to read out the nucleotide triplets, 3 letters in the mRNA sequence, and associate that with a particular amino acid. And so this is where Crick's adapter hypothesis came in. And this was a hypothesis. This was not the only way to solve this problem. One way that one could have anticipated that the mRNA message could be read would be that there would be a physical association between one of the amino acids and a particular repeat nucleotide triplet. But in fact, that's now, we know, is not what happens. Instead, there is a separate adapter molecule that provides the physical interaction with the nucleotide triplet, which means that someplace else on that molecule, we need to physically associate the amino acid. And what we're going to talk about today, really focus on, spend most of our time on, is how to physically associate the proper amino acid with the proper tRNA that reads the proper nucleotide triplet. And this is like a really essential nuts and bolts aspect of the genetic code. And just like so many other things, if you need to get a tough job done in an accurate way within the cell, you're going to call upon an enzyme to do the job. And we're going to talk about a whole family of enzymes, the aminoacyl tRNA synthetases, that are responsible for doing this job of placing the right amino acids on the right tRNA adapter molecules. Yes, sir?
Can you reiterate the difference between Crick's hypothesis and what we actually see happening?
What we actually see happening is Crick's adapter hypothesis. Crick hypothesized that there must be an adapter molecule. That's in fact the case. And that adapter is the family of tRNAs. Great. So here's our genetic code. And one of the things that we can ask you is what triplet codons are assigned to each of the 20 amino acids. Right, need to get that fixed. 20 amino acids. We're not going to ask you to memorize this table. We're going to ask you to work with it. We're happy to give it to you. You might want to know that UGA is a common stop codon or something like that. You should probably know that AUG is the start codon. And we'll talk about that, not so much today, but on Thursday. The beginning amino acid for the beginning of any mRNA message that's read out into protein is always that AUG that encodes for methionine. And we'll see how that plays out when we talk about the initiation of protein synthesis. But other than that, you don't need to memorize this thing. You just need to be able to see an mRNA sequence and be able to say, okay, ACU, threonine, GGU, glycine, and be able to construct the polypeptide, specific polypeptide sequence from N-terminus to C-terminus given an mRNA message. So we're not going to ask you to memorize this. What I'm more interested in is making sure that you know how this information was obtained. And think about it this way, like, this is only one possible way we could have created a genetic code. Here it is, you can use it, no reason to memorize it, but we just landed this Explorer probe on the surface of Mars. Like, what if we found some new life form there, and we know that, found out that, yeah, it's got a particular informational genetic molecule. I would want you to be able to describe to, a funding body what experiments you would do to crack that new genetic code. That's the kind of mindset that I'd like you to have. Not memorizing the one that we have, but being able to figure out how to describe a new one. And the other big thing that I'm going to ask you to know as we move through this material is, how does the cell get this right? How does it make sure that every time there's an AAA, it puts in a lysine, but when it sees an AAC, it puts in an asparagine? How does it get it right? So those are the things that I care about, and I hope you will care about them too. So let's talk about the genetic code, right? We're going to be working as a template with a particular mRNA molecule. Let's figure out how to read the information that's in that molecule. And the first thing that we need to figure out is whether it is an overlapping code or a non-overlapping code. So I've given you the information already that we're going to be looking at nucleotide triplets, right? So here's 3 letters. That's going to code for 1 amino acid. But then, all right, so let's say that's the first bit, first amino acid. How do we look at the molecule and figure out what the next amino acid is going to be? One possibility would be just to skip to the next codon, next 3 letters, and read that one. But we could just move 1 letter down and then read the next 3 and the next 3 in an overlapping way. Moving 1 at a time, reading 3 letters at a time. And what we know is that it is a non-overlapping code. This is the way we read it out. We read first 3 letters, then the next 3 letters, and then the next 3 letters, and we move down the mRNA molecule that way. So it's a non-overlapping code. It's successive. We don't skip. We read every letter. Once we start at the start codon, we read every letter in this non-overlapping way. The other thing that we typically say about mRNA messages is that there's no punctuation. What do you think that means, that there's no punctuation? And you can look at these issues as though you're trying to figure out a new language, right? Somebody hands you something in Cyrillic or cuneiform. How do you read the message? What does it mean there's no punctuation? Yeah. Exactly, right? So here I've told you that here's the first triplet, and once that's defined, then, and we know it's not overlapping, then I can figure out, okay, here's the next triplet and the next triplet. But if you didn't know that, if you just were handed a piece of mRNA, there's nothing in here to tell you where the separations between the triplet codons are established. It would be kind of nice if we had commas, like AUA, comma, CGA, comma, but there's nothing in the molecule to provide that kind of punctuation. So we need some other kind of strategy to be able to figure out what the reading frame is. And that term reading frame, I'll get back to in just a second. So no punctuation, we read 3 triplets at a time in a non-successive, in a successive way, non-overlapping, but we need to figure out which are the meaningful codons. So reading frames, right? Once we define one sense codon, we can begin to move down the molecule and read out all the others. But for a particular mRNA molecule, it's possible to imagine being able to read multiple different reading frames. Right? So if there was some kind of ambiguity, and in some circumstances there is, we could start at this U, or start at that U, or start at that C, and just read in these different reading frames, reading out different triplets, which would presumably create different polypeptide sequences. And we'll look at some experiments in which that's exactly the case. Ribosome gets a little confused, doesn't quite know where to start, and begins churning out 3 different messages in these 3 different reading frames. Does that make sense? Reading frames? Makes sense? Okay, great. Because that'll be important when we begin looking at these experiments that solve the genetic code. All right, so I'm going to describe to you now 3 different types of experiments that were used back in the early days when they knew that mRNA provided the message, they knew that the protein was the product of the message, and it became an issue just trying to figure out how to fill in the genetic code. So the 3 main strategies were in vitro protein expression with a random polymer of mRNA that was made by polynucleotide phosphorylase. And I'll explain that in a second. Binding studies of amino acyl tRNAs to ribosomes in the presence of specific mRNA. And in vitro expression with defined repeating sequences of mRNA. So let's talk about what this means in vitro expression, right? Because that's common to 2 of these strategies. It just means that I bust open a cell, and I take the ribosomes that are sitting in there, and I put them into a test tube with a bunch of goodies. The amino acids, ATP, salts, everything that's needed to make sure the ribosome can do its thing. All right? So that's an in vitro expression, and then we can dribble in there a particular mRNA molecule and just see what the ribosome churns out. So it's taking the machinery out of the cell and creating an artificial system in which the ribosome can do its work. And there's just tons of cool experiments that you can do with these cell-free or in vitro expression systems. So in one case, the experiments involved generating a random polymer of RNA. In another case, more sophisticated experiments were done in which they provided very specific sequences of mRNA to these in vitro expression systems. So let's talk about this crude method first. So this was the first thing that they were able to do is just use this enzyme, polynucleotide phosphorylase, which just assembles, you just feed it nucleotides, and it'll just put them together in a random kind of way. So it's kind of making a sort of a nonsense message, but you can actually tease some information out of it, because let's say that you just fed it adenines. Well, then you'd kind of have a fairly well-defined polymer. It would only be adenine. Or you could have very specific ratios between adenine and, let's say, one other nucleotide, in this case, cytosine. So that's the kind of experiment that was done, is that they knew the relative ratios of all the different codons that could be made from those 2 nucleotides. And if they defined the ratio, like A to C is 5:1, then they could come up with an expected frequency of incorporation of any particular nucleotide. So they would know that, if you're mostly feeding this system adenine, then probably most of the codons that will be read out will be AAA. And what they were able to observe is the majority of the product that was being created was a polymer of lysine. So right away they are learning information about the fact that lysine is presumably encoded by that particular triplet, AAA. But if you take a look at all the other codons that could be made from those two nucleotides, clearly asparagine, glutamine, histidine, proline, and threonine are also encoded by those other nucleotides, for example, AAC or ACA, etc. So they can calculate what the expected frequency is going to be versus the observed frequency. So this gave them some information just based upon a random incorporation of different defined ratios of nucleotides within the mRNA. Okay. A little bit crude, gave them some information, not a huge amount, but was a great starting point. And this is what the experiments looked like. They're conducted by a couple of guys, I think they were at the NIH, National Institutes of Health, Nuremberg and Mattei, and this is what their data looked like. They were associating different triplet codons with incorporation of different labeled amino acids. And again, no need to memorize anything like this, just understand how these experiments were conducted. So here's a particular example. I don't have the ability to make a really defined sequence, but for sure, one of the things that I can do is use this polymerase method to make just this stupid mRNA, just repeat U over and over and over again. So what does the codon UUU encode for? We're going to create a whole bunch of test tubes. We're going to put our cell-free expression system in there, all the ribosomes, all the substrates that they would need, the ATP, etc. And we're also going to include different amino acids in each of these test tubes. We're going to add the same RNA to each of these. And in one case, and in only one case, will the ribosome be able to polymerize those amino acids and make a protein chain. And the only case in which that ought to happen is the case in which the UUU message matches up with the actual amino acid that it codes for. And when Nuremberg and Mattei did that experiment, they found only one of these 20 tubes actually created a protein polymer. That was the tube containing phenylalanine. Only created polyphenylalanine in the presence of this poly-U RNA, and that told them, yes, UUU is the codon for phenylalanine. Yes, sir.
So if you did these in ratios and then you found out that, hey, you wanted to mix up like A, U's, and G's in equal ratios, then how would they tell apart, because the ratios for A, U, and G would be the same? Like, how did they tell apart when was the start of the frame?
Fantastic question, and the answer is they cannot. They cannot tell them apart, and that's why right here, these tentative assignments, you see that's an A2C. They cannot discriminate between ACA, AAC, CAA. They can't tell the difference. And that's why I'm going to talk to you about 3 different experiments that were used to solve the genetic code. All they were able to do then is say, all right, UUU, phenylalanine, but we can't assign any of these others yet because they weren't able to create specific mRNA messages at that point. There was another question. Yes?
Why doesn't the UUU mRNA require GAG circle for translation?
To be efficient, it actually does. So it's kind of amazing that these experiments worked at all, but there's some baseline rate at which the ribosome will just sort of get itself started. But yeah, all of these things technically should require a start codon, absolutely. And in my graduate biochemistry class, we actually go and read these papers and we find out that, it's really easy for us to say these experiments worked every time, But when we look at the data, we see that sometimes these issues about like not having a start codon really had a huge influence. And you can imagine that when they finally made specific mRNA polymers, the one that does have AUG, that worked like gangbusters, right? To create this polymethionine, that was phenomenal. They had trouble understanding why a poly-AUG substrate template all of a sudden like just cranked out the protein polymer beautifully. Why is that one so much better than anything else? Well, now we know that's the start codon. Okay, other questions about this type of experiment? Great. Experiment number 2, ribosome binding experiment to radio-labeled amino-acyl tRNAs. Right. So we will see in really great detail on Thursday how the ribosome is organized so that it accepts tRNAs and associates the anticodon loop, looked at that before, with the codon on the mRNA. And there's 3 particular places within the ribosome that different tRNAs can sit at different stages in the process. Really, the important thing that you need to know now is that the ribosome is like a stage upon which the template mRNA is matched up with the proper tRNA. And that physical association provides us with the opportunity to really tease this kind of thing apart. We can take a ribosome, associate it with a particular mRNA, and just ask which amino-acyl tRNA is bound. Right? And if we radio-label these amino-acyl tRNAs, the charged tRNAs with the proper amino acids on them, only in cases in which the mRNA is sitting on the ribosome will the tRNAs bind, and you'll take a fairly small species, the tRNA, and you'll physically associate it with this massive ribosome particle. You can just ask, for which trinucleotides will you get the tRNA to sit down on the ribosome and associate in a very stable way? These experiments were also conducted by Nuremberg with another individual later working in Nuremberg's lab. And this cartoon here describes the experiment in a little bit more detail, right? So we're going to be working with, let's say, radioactive glutamic acid in this first case. And we're going to associate it with the proper tRNA for glutamic acid, and that happens to have this codon CUC. And then we're going to ask the question about, is glutamic acid encoded by this synthetic mRNA with this particular codon GUU? Right? So we load that mRNA onto the ribosome, and we just ask the question, is there a binding reaction here? The answer is no. This codon, anticodon don't match up. There's no Watson-Crick hydrogen bonding complementarity. There's no effective binding, and as a result, this remains essentially a small species that goes right through a filter and is not bound onto that filter. Now let's repeat that experiment with a radioisotopically labeled valine. And we're going to put that on its cognate tRNA, which happens to have the anticodon CAA. Now we've got a great match with this GUU synthetic mRNA. And when this is sitting on the ribosome, that tRNA goes, okay, found my complement. I'm going to bind into that ribosome, and now I'm part of this massive ribosomal particle. And now, when I pass that on a filter, that whole thing is going to be trapped onto the filter because this thing is so big. So in this case, the radio label, the radioactivity goes into the filtrate, and in this case, the radioactivity gets bound onto the filter. And you do this one at a time for a whole bunch of synthetic mRNAs, and just ask the question, which radioisotopically labeled aminoacyl tRNAs are bound in each case? And this works pretty well. It allows you to flesh out a little bit more of the genetic code. Questions about this experiment? Yes?
How did we know what class of tRNAs were involved in the first place? Like, I understand how the that hypothesis was established, but did they separate out the entire splicing?
Yeah. I'd have to go back and actually read these papers to find out how precisely they knew what the aminoacyl tRNA synthetases were doing, whether they were working with purified material that they had created in vitro, or whether or not they had just somehow managed to purify the properly charged tRNAs from the cell extract. I don't know the answer to that. Yeah. Right, so what information did they have going into this experiment? They had some of the information from these sorts of experiments, so they had some of the codons figured out, so they could validate this experiment, right? This experiment told them that UUU encodes phenylalanine, so they were able to use this to establish that this technique worked. And so in this case, they were working with a tRNA for phenylalanine with the phenylalanine on it that was labeled with a radioisotope, and they found that in the presence of that UUU trinucleotide, it was bound onto the ribosome. For a few of them, they knew, like these pure repeating ones, they knew these codons and they were able to validate the experiment. But like for ACA, for example, they didn't know what that encoded for yet. And it's, yes, in this second experiment here, they didn't know what it encoded for yet. They knew a few of them, not very many. They used the information from the ones that they knew to validate that this experimental approach worked. And then they ran through it one at a time with these different synthetic mRNAs to find out in which cases the corresponding tRNAs were associated with the ribosome. Yes?
Sorry, so just to repeat, they were only able to find which amino acids were coded for by 3 mRNA codons that were like the same, like either they couldn't find.
Right. So here's our first experiment with random message. They were able to definitively associate only when they had these repeating nucleotides. They knew those for sure. In other cases, there was ambiguity. That was from the first experiment. From the second experiment, in some cases, they were able to confirm the initial assignment and to create new assignments for these ones that were other positions in the genetic code, such as GUU. Yes?
So getting the same results for the ones that they were able to determine with the first experiment means that the second experiment works.
Exactly.
And then you can do it for the other.
Proof of principle here. Experiment works. Now we can move on to evaluating all the rest of the codons. And this experiment worked and fleshed out a lot more of the genetic code, but not all of it. So a third experiment was conducted. And this was done by one of my scientific heroes, this guy Gobind Khurana, who was a father of oligonucleotide synthesis. He was a phenomenal synthetic chemist, and he figured out ways to synthesize DNA polymers in specific sequences. And you know, I'm sure a bunch of you at this point have already requested specific DNA sequences. You just, send out this form for a particular primer. It comes to you the next day. You specify 20 base pairs, 30 base pairs, no big deal. Boom, you have it. Man, trying to make these things in the early days of bioorganic chemistry, this was super painful. Khurana figured it out. And because he could make specific DNA sequences, he figured out how to transcribe them into particular RNA sequences, and now he had specific RNA sequences that he could feed into these cell-free protein expression systems, and that was really the advance that allowed them to completely crack the genetic code. All right. They got a huge bunch of information because what Corona could do was make an mRNA that was UUC, 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 UUC over and over and over again. Not some random assembly of U's and C's, no, specifically UUC over and over again. And then they just fed these into these cell-free protein expression systems. What polypeptide is turned out? Oh, well, they got phenylalanine and polyserine and polyleucine. They got 3 different products. So I told you that, like, he's able to make a specific RNA message. There's still some ambiguity. Why? Why did he get 3 different products? Because of the reading frame, exactly. So yeah, you can make UUC, UUC, UUC. The ribosome's going to jump on that, and it's going to begin cranking out the protein message, but it doesn't know where to start because there's no punctuation and there's no start codon. It'll manage to start, but where does it start? Does it start and begin reading UUCs, or does it read UCUs, or does it read CUUs? There's nothing to tell it. There's no punctuation. And it'll make all 3. And that's what they found. Polyphenylalanine, polyserine, and polyleucine. But move through making all these different things. Some cases, he's making 3 different products. In other cases, he's making 2 different products. He also was able to make tetranucleotide repeats, right? So UAUC repeated over and over again. Obviously, this can be read out in different reading frames. In some cases, they got just di and tripeptides. They weren't getting polymers. They were feeding a polymer message, but only getting little short guys out generated. Why do you think that they were only getting little short guys? Yeah. Stop codons are embedded within these things. Exactly. So it puts a couple amino acids together, then sees something should be stopped, quits right there. So here's the guy, Golpanian Corona. This guy came from unbelievably humble roots in India. I think he worked at the University of British Columbia and then the University of Wisconsin and ended up in MIT, if I'm not mistaken, and he died only just a few years ago. And this really illustrates why one defined message gives you 3 different products. It's because the reading frame in these cases is ambiguous. Now, when I hand you an mRNA sequence and I tell you what product is going to be made from there, most of the time, somewhere in there, I'm going to embed a start codon, an AUG, and you'll be able to see that and go, okay, that's where the ribosome is going to engage, and now I've defined the reading frame. I read out that start codon, which encodes for, what did I tell you? Thymine. And then you'll just read successive triplets. So even though there's no punctuation within that mRNA message, you'll be able to figure out the unique product that's going to be generated. Okay. This is a little bit more reiteration about why a particular synthetic polynucleotide provides 3 different messages. Just repeats the same concept. All right, so this describes why, in some cases, these repeating tetranucleotide sequences would generate only, let's say, a di or tripeptide. So if we repeat the sequence GUAA, 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 and ask what is created by that particular message, you'll appreciate, okay, 3 different reading frames. We can start here. I'm going to read out that, read out that, read out that. Now when I get to the 4th codon, UAA, that turns out to be a stop codon. So I can make a tripeptide. There's no tRNA that's going to read that message. So it's going to stop and just make a short little guide. And it'll do this over here as well. It'll be reading out GUA, AGU, AAG. It'll hit that UAA again and stop. After all of these experiments were completed, the 3 different types of approaches, work in the Nuremberg lab, work in the Corona lab, they were able to flesh this out. It took them a little while to really understand that there were 3 stop codons. Right? They were fleshing this thing out. There were big holes. And then, okay, we got everything filled in. Still haven't assigned these 3 codons, UAA, UAG, UGA. They probably are stops. Why do we need 3 stop codons? There's no good reason. Theoretically, you only need 1, but in fact, there are 3. But this is what they finally settled on. Yeah.
Do we have any idea as to why that's like AUG is the start codon and like UAA, UAG, and UGA are the stop codons?
You know, I'm going to answer that the way that I would most questions about why the genetic code is this way. Totally arbitrary. Why is methionine, and it's actually a little bit of an altered methionine, why is that the amino acid that the cell uses at the start of every protein chain? I don't know. It's so easy to contemplate ways to rewrite this code. It's arbitrary. And in fact, a lot of work that I did in a previous life, we've messed with this thing so bad. We reassigned tRNA anticodons. We rewrote this thing and got the cells to crank out stuff that biology, living systems never imagined. So there's no reason why it has to be this way. And the proof of that is you can rewrite this. And maybe if I have time at the end of lecture today, I'll show you some experiments that I did to demonstrate that. Yeah?
Do you have like a biological dysfunction where your ribosome just doesn't stop when it sees a stop codon?
Yes. Stop codons are not always properly recognized. I don't have an example of that. These are often in the case where a virus is got a message and it will skip. It will called a frame shift. Yeah, it won't read that stop codon properly. And here we are. Altered reading frames. Right? So, okay, this is for a Rouse sarcoma virus. And the anticipated reading frame is going along. Okay, we know the start, and it's coming along to the end of this particular gene. And leucine, glycine, leucine, arginine, leucine, threonine, asparagine, leucine, and then we hit our UAG. That's a huge stop codon. And that's where the message generally ends. So this isn't quite a disease state, but this is an example in which there's a frame shift. And instead of the ribosome seeing that stop and reading it properly and disengaging from the message and terminating synthesis, instead it hiccups and it backs up 1 and it says, okay, I could read that UAG, but instead I'm going to just go back 1 nucleotide, and now I've got another sense codon. And I'm just going to read that as AUA, isoleucine, and now we're back to the races again. So this is a frame shift, and this allows 2 different protein products to be made by this particular gene in this virus. There's another way in which you can think about a disease state being associated with a stop codon, Imagine a mutation here, right? And it should be a stop, but we've mutated this so that it actually has a sense codon where there should be a stop codon. Those provide a range of, in some cases, very severe genetic diseases.
Would you, in that case, do you mutate the G so it's no longer UAG and a stop codon, or it's still UAG, just not a stop codon?
Oh, it's not UAG. There's a mutation to some other triplet. Yeah. Okay. Almost every time I'm going to show you like a mature mRNA, and that's it. That's what we use as a message. There are cases in which mRNA can be edited to convert one residue to another. Those are just real oddballs. You need to know that, okay, it could happen, but it's a pretty rare thing. The genetic code is degenerate. Right, so we know there's 20 amino acids. We know that we can create 64 different triplet codons. We're going to go ahead and assign 3 of them as stops. Like, why? 3, why not 2, why not 4? No reason. 3 are stops, so we get 61 sense codons. 61 sense codons for 20 amino acids. There's multiple triplet codons that encode for the same amino acid. And in some cases, it's kind of ridiculous. 6 different triplets encode for arginine. Why? 6 different codons encode for serine. 6 for leucine. Why? I don't know. Tryptophan and methionine are the only ones that are encoded by 1 codon. I guess that makes sense. Tryptophan and methionine are actually fairly rare. Tryptophan is the most infrequently used amino acid, so maybe we only need 1 triplet. So only 2 are encoded by 1 unique codon. The others, we're looking at a degenerate code. All right, so what's the physical chemical basis for the way that we can look at a triplet and associate it with a particular amino acid? Here's our adapter molecule, tRNA. We're going to put an amino acid on there. We'll get to that in a bit. But for right now, we just need to read out the codon, and we do that the same way that we do DNA replication or DNA-encoded RNA synthesis. We provide for Popper, Watson-Crick hydrogen bonding interaction. So the canonical way to read out a codon from 5' to 3' on the RNA that is AUC would be to provide the Watson-Crick pair. Reading from 5' to 3' on the tRNA. Keep in mind we're going in the opposite direction, 5', 3'. GAU is the proper anticodon for AUC. All right? This interaction at the first position, this interaction at the second position, this has got to be the right Watson-Crick base pair exactly. It turns out that this third position in the codon, the first one in the anticodon, this is, you can get it kind of right. There's a little bit of, I don't know, you can play around there a little bit. It tolerates imprecision here. And so we call this a wobble interaction. And this is a position in which we can tolerate non-Watson-Crick base pairing. And so now we're beginning to see, like, okay, well, maybe that is one way in which multiple different codons can be associated with the same amino acid. And here's some examples of that. Here's a nice illustration from your textbook showing a wobble interaction between a codon and the anticodon loop on a tRNA. Again, exact Watson-Crick base pair at the first 2 positions, but here we can have in the codon either a C or a U, and in both cases it's going to be read by a G that's present in the anticodon of the tRNA. Both of those bind sufficiently well that that same tRNA will associate with 2 different triplet codons. So this gives some examples of these different wobble interactions. And in every case, the variability is going to be at that third position. Right? That's the wobble position. Here's a crazy example where a modified nucleotide, inosine, it actually forms a good enough interaction with 3 different nucleotides. So the same anticodon with inosine in it reads out 3 different triplets. So that's the most extreme example of a wobble-based interaction. All right, so that is the genetic code and the way it's read out. And is it hard and fast? Is it 100% of the time? 99% of the time, that's the way that we read these things out. There are exceptions to the universality of the genetic code. Personally, I am astounded that it doesn't matter whether you're talking about humans or blue whales or microbes from a deep sea vent or a panther or an ocelot or a sloth, the same genetic code applies. And, doesn't matter, simple organisms, complex organisms, that genetic code, you can take it to the bank, right, and it's going to apply in 99% of the cases. There are some variations in codon assignments, and sometimes these pop up in mitochondria, where something that you would expect to be read out as a stop actually will provide a second triplet codon for tryptophan. So in this case, in the mitochondria, there isn't just one codon, there's 2. And this indicates that you can actually read out something that would be a stop codon and turn it into a sense codon. Other cases, like something that you would normally expect to read out as an isoleucine, might end up like in a yeast mitochondria as a methionine, and some other completely oddball examples. I'll just say that, does this mean that our genetic code is uncertain or really high variable or depends on the circumstances? No, you take it to the bank, right? These are just exceptions that really prove the rule. That genetic code is pretty firm. But the exceptions are kind of fascinating. So here's a 21st amino acid. We think of there only being 20, but in total oddball cases, nature calls upon proteins that incorporate this really weird amino acid. This is selenocysteine. It actually looks a lot like a serine or a cysteine, but it's got a selenium where the oxygen or sulfur would be. And this actually is read out in response to what you would normally think is a UGA stop codon. This is created by a serine loaded onto a tRNA that recognizes that stop codon. And then that whole complex, there's an enzyme that goes in, pulls off the hydroxyl group, replaces it with selenium to make this really bizarro amino acid, selenocysteine. And I should also mention that there's a 22nd amino acid that's also been described within the past 10 or 15 years, this pyrrolysine. So that's a really unusual structure. You'll almost never see this come up, but again, these are exceptions that prove the rule. All right. Any questions about how we read the genetic code? Yes?
So the previous slide, that 41st amino acid, is really unique functionality that is useful?
Yeah, that's selenocysteine. It's used, and I think it's in an active site of an enzyme where it's really, you need that selenium there. And that's why selenium actually is a necessary nutrient. It's one of these really bizarro nutrients that need just a tiny amount, and if you take too much, it's actually really toxic. But the reason that we need selenium is precisely because of that. There is an enzyme that requires it. Yeah.
I think you need mRNA usually read
Oh, yeah. Are the same genes read out in different reading frames in different tissues? I think so. Yeah.
not just the splice forms, but the actual
I'm gonna say yes, but I'm also not going to be able to provide you with any specific examples, but I think so. Okay, so let's get into the mechanics of protein synthesis. We're gonna make them on ribosomes. We're not just going to be tossing individual amino acids at the ribosomes. That actually wouldn't be energetically reasonable. Amino acids don't like to be polymerized. They like to be, polypeptides like to be hydrolyzed. And when I say that, I mean it's energetically favorable for them to be hydrolyzed. So we need to create amino acids in an activated, chemically activated form. And we do that at the same time that we associate them with the tRNAs. On Thursday, we'll talk about initiation, elongation, and termination of polypeptide synthesis on the ribosome. And I want you to understand all those steps. And then we'll also see that for a given mRNA, the ribosome can read through, get to the end, stop codon, we're done, disassociate. That same mRNA can be used by the ribosome again to encode the same protein message. Here's a picture of the ribosome. We're going to be talking about 5 different steps. We're going to be talking today about activating the amino acids associated with tRNAs. Then on Thursday, we'll talk about initiation, elongation, and termination of the polypeptide chain on the ribosome. We've kind of already talked about folding and post-translational processing, but we'll review that again, especially when it comes to post-translational modifications of the protein message. Okay. The ribosome is super complex. We don't need to get into the details today. We'll do that in fairly painstaking detail on Thursday, but massive piece of machinery. 2.7 million Daltons, and you know, occasionally I've described to you guys like cases in which have been completely overwhelmed with the scientific discovery. This was one of them when they provided the X-ray crystal structure of the ribosome. I thought that there were limits to structural biology. I thought that there were some pieces of machinery, some proteins that were just so complex, they were never going to be able to use X-ray diffraction techniques to solve those structures, but this was truly extraordinary. They have an atomic level resolution structure of the ribosome, and now we are using it to really figure out what the ribosome does. Again, we'll take a look at that on Thursday. Protein synthesis occurs in this cleft, and we'll take a look at some of the details there. Really, the one thing I want to stress, because this is also just totally freaks me out, and so I'm going to mention it today, and I'll really hammer on this on Thursday. The ribosome is a ribozyme. Does anybody know what that means? Anybody figure that out? That just got me going. So I told you guys that RNA could be catalytic. It could act like an enzyme. Right? You know, the self-splicing and that kind of thing. Your textbook has some other examples of the hammerhead. We call those catalytic RNAs. We call them ribozymes. They're ribonucleic acid. They act like enzymes. We call them ribozymes. So now that I've told you what ribozyme means, what does this sentence mean? The ribosome is a ribozyme. Yeah.
Catalyzes its own production.
No, it does not mean that the ribonucleic acid catalyzes its own production, because this piece of machinery cranks out protein polymers as a product. So then what do I mean by this? Yes. It's a non-protein enzyme. Wow. It contains both protein and RNA, but the enzymatic part of it is the RNA, not the protein. Could not believe that. Still have trouble accepting it, but we'll take a look in detail on Thursday, and I'll convince you that that's true. All right, so here's our tRNAs. This is what we're going to be feeding to the ribosome. We took a quick look before at the anticodon. All the tRNAs have that, these three positions that are going to associate with the mRNA nucleotide triplets. Here's our wobble position right here. These two positions are exact match. Here's our wobble. And these sorts of things we see in all tRNAs. The D arm, this TC arm, and then there's the amino acid arm. It begins, or actually it ends with this CCA to the 3'. And here is where we attach our amino acid. Right? So here is where we hit the mRNA. Here's where the amino acid is positioned for every tRNA. And there are a family of enzymes, the aminoacyl tRNA synthetases, that are responsible for conjugating the amino acids at that position. Here's the structure of the tRNA, three-dimensional structure now showing this is the anticodon right down here, and here's our acceptor stem, CCA. Here's where we need to attach our amino acid. All the tRNAs are about 80 nucleotides, give or take a little. I mentioned before that a lot of the bases are modified, including oftentimes this inosine that's present in the anticodon. And they all form this cloverleaf structure that you can see in a two-dimensional representation. And in the presence of the divalent cation, it then forms this three-dimensional structure right here that looks more like an L. All right. Here's the enzymatic process by which we're going to associate the proper amino acid onto these tRNAs. It's done by the aminoacyl tRNA synthetases. And this is a family of amino acids because there's a separate protein for just about every one of the 20 natural amino acids that we're going to be dealing with. We know the structures for these enzymes. We have a pretty good idea of their mechanism. And this is the chemistry that they do. So first, they're going to activate an amino acid by attaching it to an adenine individual nucleotide to create what's called an aminoacyl adenylate. So here's the reaction. This is the nucleophile, the carboxylic acid group, nucleophilic attack on the alpha phosphate of the 5' triphosphate group of an adenine triphosphate. Come in, ATPs are the substrates. Here, an amino acid corresponding to whichever aminoacyl tRNA synthetase, the proper amino acid, performs this nucleophilic attack and creates this product. Different R groups for the 20 different natural amino acids, but in all cases, these aminoacyl adenylates. And this is actually a highly activated species. This is an anhydride. And you can check out this structure at home, convince yourself it's an anhydride and is a highly activated species. Now that we have this activated entity, these different aminoacyl adenylates, now the enzyme attaches it onto the proper tRNAs. And here things get maybe a little bit complicated. There's 20 of these synthetases. They fall into two categories, two classes. And the mechanism is slightly different. For these group 1 aminoacyl tRNA synthetases, we're going to initially attach the aminoacyl group onto the 2' hydroxyl group of the adenine that's right at the 3' end of the tRNA. Right? So the enzyme needs to grab the right tRNA. It's going to go onto that acceptor arm, that CCA. That A is the last position, the 3' base in every single tRNA, and it's going to use that 2' hydroxyl nucleophilic attack on that anhydride group to form the amino-acyl conjugate at that 2' position. And it releases AMP as another product. For the class 2, does the same exact reaction, except it uses the 3' hydroxyl as the nucleophile to create the amino-acyl tRNA at that 3' position, again releasing AMP. In every case, the ribosome uses an amino-acyl tRNA that has the amino acid at that 3' position. So for these class 1 enzymes, it just needs to shift that amino-acyl group over from the 2' to the 3' position. A little bit complicated. End product is always the same. Initial amino-acyl adenylate, also always the same species, with the 20 natural amino acids. So these are the products of this enzymatic reaction. These amino-acyl tRNAs created in 2 steps. And you should be able to sketch these out. So here is the products after the first step. Right? These amino-acyl adenylates, an anhydride group, 20 different ones are created with different R groups corresponding to the 20 different natural amino acids. Here are the class 1 amino-acyl tRNA synthetases. Here are the class 2. Turns out there's 10 and 10. Why 10 and 10? I don't know. Nobody really understands why there's 2 classes of amino-acyl tRNA synthetases. There's some speculation. The structures of these 2 different classes are pretty much different, class 1 and class 2. It's interesting to see how the tRNAs are engaged by these enzymes. Again, remember, these enzymes are providing a critical role in establishing fidelity. They've got to make sure they grab the right tRNA and attach the right amino acid at that CCA arm. So it needs to be able to hug these tRNAs and make sure it's hugging the right one. Where is it engaging them? Well, the anticodon is a really good place to discriminate one tRNA from another, right? Those have got to be different for the different tRNA species. But there's other positions of variability for the different tRNAs. There's about 30 to 40 of them within the cell. Right? So this shows you that there is multiple points of contact, including the anticodon, including the CCA arm, and this is the point of business, right? This is where the catalysis is going to occur, because this is where the amino acid needs to be attached onto that adenine, forming that amino-acyl tRNA at that adenine position. So active site here, active site here. This is what the chemical structure is that's generated by these enzymes. Right? Here's the tRNA, the 3' end, CCA. Here's our adenine, 3' position as bearing the amino-acyl group, esterified at that 3' oxygen. So this is the way the adapter molecule works. This particular anticodon is matched up with a particular amino acid way on the other side of that molecule. How does it get it right? We've talked about this before. In some cases, different substrates have got a lot of structural similarity. Here is valine. Here is isoleucine. They have a lot of common features. What's the difference? One methyl group. It's asking a lot of an enzyme to grab this guy every time and not be kind of sloppy and grab this one from time to time. And you can work out the energetics, like how much energy would you get by binding to a methyl group, and you can convince yourself that there's no way that these enzymes are going to be able, just from a thermodynamic standpoint, of getting it right every time. So just as in the case of DNA polymerases, where I told you that there's a proofreading mechanism, these enzymes, which have to get it right a lot of the time, they also undergo proofreading and editing. There's a two-stage process where they make an initial guess, and they go back and say, did I get it right? And if it got it wrong, it cuts it out and starts over again. So editing functions exist in many aminoacyl tRNA synthetases, and there's different ways that they perform this editing step. We won't get into the nitty-gritty, but I'll tell you that the final error rate is about 1 in 10 to the 4, and that's pretty darn good. And it's important that it needs to be that good, because you can calculate, like, if the frequency was sloppier, you know, let's say it was right 1 out of every 100 times. Well, if you were making a 100 amino acid protein, you'd be making something not quite right a lot of the time. You'd only be getting it absolutely correct about 37% of the time. But this is a really short protein. The cell makes 300 amino acids, 1,000 amino acid proteins all the time. And if it wants to get it right, like 90% of the time for these larger proteins, it needs to be operating at about 1 error in only about 10 to the 4th positions. So fidelity is crucial. And as we take a look at these enzymes, we'll see that the activation site and where it conjugates the amino acid onto the tRNA is just one of 2 active sites in the enzyme. There's also a separate editing site where it can go and check to make sure it's getting it right and cleave it off if it hasn't. So this is a 2-step process. We call it a double sieve. The active site has got the same type of complementarity for its proper substrates that we've talked about a lot in the case of enzyme catalysis. And so that's kind of like a first sieve, right? It's looking for an amino acid that's like hydrophobic and about a certain size, and that's what comes into the enzyme active site. Then after it's performed that conjugation, it'll actually push that product over into a neighboring editing site, and that site will look at the product and say, does that seem correct? Is that the right shape? Am I seeing an extra methyl group that I'm not expecting? And if it is incorrect, like in this case, a valine has been added onto a 3' tRNA, that editing site will be able to recognize that error, goes, that's not right, and it'll just cleave that amino acyl group off that 3' position, giving us back the amino acid, giving us back the tRNA, and it'll start the process over again. So this is this double sieve mechanism for ensuring fidelity. Now, is this the only way that the cell makes sure it's correct? So what would happen if we took the wrong amino acid, put it on a tRNA, and sent it over to the ribosome? Is there an editing capability on the ribosome? We can do that experiment. Here's an example. We use one of these synthetases to do exactly what it ought to do. Give it cysteine, give it ATP, give it the proper tRNA, boom, it adds the cysteine onto the 3' position to make that amino acyl tRNA. Correct. Cysteine conjugated onto the cysteine tRNA. But now we bamboozle it. And we treat it with rainy nickel in the presence of hydrogen gas, and we reduce that position and remove that thiol group, and now we've converted that to an alanine. And we've got a completely different amino acid on the same exact tRNA. We've got alanine conjugated to a cysteine tRNA. And you take that and you feed it to a ribosome. The ribosome goes, I don't care. I'm going to make the proper association between the anticodon loop of the tRNA and the mRNA, and I'm just going to put on whatever amino acid is on the other end of that tRNA. And when it sees a cysteine codon, boom, in goes malonine. The ribosome does not do error checking. So that's why these amino acyl tRNA synthetases are so important and why we call them the second part of the genetic code. So yeah, back to this question, why does the genetic code, why is it that way and no other way? Man, we can mess this thing up so bad. So this is work that I did before, where we took the amino acyl tRNA synthetase for phenylalanine, and we just we fed it these like bizarro amino acids. Like we fed it these guys, no problem. The amino acyl tRNA synthetase never sees these things in the cell. It doesn't have the capacity to recognize these things as being ridiculous, and it just puts them on the tRNA. Boom, makes a protein. Where there's a phenylalanine codon, in goes these guys. And then we made additional mutations in the amino acyl tRNA synthetase so that we opened up the binding pocket, and it was accepting all of these bizarro amino acids, putting them in right in for the phenylalanine codon. The ribosome doesn't check. Ribosome is just looking for codon-anticodon interactions. Phenomenal chemist Peter Schultz scripts in La Jolla, California. Look at these wacko amino acids. Ribosome doesn't care. You'd almost put a brick onto the end of an amino acid, and the ribosome would still accept it. All right, we'll talk about the process of ribosomal-mediated protein synthesis. For today, the summary here is know what goes into a mature mRNA, understand the adapter hypothesis, know those 3 experiments that crack the genetic code, know the reactions that are catalyzed by the synthetases, understand the second genetic code specified by the amino acyl tRNA synthetases, and this double sieve mechanism. And I'll give you a break and I'll talk about the densest experiment on this.
All you do is stop it, and then just click the button.